This podcast will serve as an introductory podcast or a guts module for the cell and tissue biology course. And it's just a brief introduction. It's a review of things you should already have had or are familiar with. This is not a formal lecture topic for our course. You should be able to explain principles of the cell doctrine, be able to explain why cells share a common evolutionary history, and clearly be able to describe differences between prokaryotic and eukaryotic cells. Now, for our course, you should be able to describe the structure and function of the major intracellular compartments and organelles within eukaryotic cells. We will spend some time over the next few days reviewing those for you. You should be able to identify organelles and subcellular structures on electron micrographs, and this is an overarching learning objective for our course. You might want to start becoming familiar with how to use a light microscope. We have a podcast on iTunes that can help you with that. Now, this is kind of a simple-minded slide, but I like to put it in to kind of remind you of what you have done so far in medical school. This image up at the top left, and then the next one over, you could think of that as gross anatomy, where you're looking at overall gross structures. The images here, where you're seeing an epithelial sheet, where you're seeing a low-power electron micrograph and maybe a higher power electron micrograph. Clearly these are diagrammatic images, but these images would represent cell biology histology, and perhaps this image, cell biology histology. The images on the bottom would reflect where you have been in biochemistry if you were trying to understand macromolecular structure. I think everyone is aware that the cell doctrine is a derivative of evolutionary theory, and I think everyone is aware of the principles of the cell doctrine. All things are composed of cells and cell products. All cells come from pre-existing cells by cell division, and all cells are similar in chemical composition and metabolic activities. I don't need to say any more about that. I think everyone understands these principles. This is an interesting way to recapitulate what we've just said. We take a lot of license as we try to put a slide like this together, but if you think of stages in the evolution of cells, you might think that the very simplest systems might have consisted of self-replicating systems based on RNA and perhaps some rudimentary polypeptides. As things evolved and became a little more complex, there would clearly be RNA-based and protein-based systems, and there'd be a flow of information basically from what's encoded in the RNA to the protein. And finally, if you could imagine going through many stages in evolution, one would have present-day cells where information is stored primarily in DNA. DNA, as you know, is transcribed into RNA, and then that information in the RNA is translated into proteins. It goes without saying that any of these macromolecular events, if you imagine them occurring in a prebiotic soup, that's one thing. But to have an efficient system where life could develop, these prebiotic things would have to be held together in close proximity. So the artist has drawn these with a membrane around them. And so clearly the origin of membranes or the development of membranes would have been critical for the origin of life. This just reminds you that cell membranes are a fluid mosaic structure. All cell membranes have a similar overall composition where you have phospholipid bilayers and proteins that float freely in the phospholipid bilayers. And you might think of these phospholipid bilayers with proteins as two-dimensional fluids where the proteins can move freely in this fluid and even the phospholipid molecules can show mobility. And we'll talk about this in a little more detail later. This is a typical example of a eukaryotic cell. This happens to be a multipolar neuron from humans, and what I'm highlighting right here would be the nucleus. Of course, you can't resolve the nuclear envelope with the light microscope. this would be where the nucleus would be, and then this very dark structure is the nucleolus. These are dendrites that would be coming into this cell body, and then the area that I'm tracing here would be the axon hillock. We'll talk more about things like this when we talk about nerve tissue later in the course. But you notice that the cell body here has clumps of heavily stained material. This clump of heavily stained material would represent rough endoplasmic reticulum, which would be termed Nissl substance in neurons, and we'll come back to that later in the course. Here is an electron microscopic view of a nucleus in a eukaryotic cell. Here, the little diagram just to put it in perspective. You know that the nucleus contains a nuclear envelope which consists of two phospholipid bilayers. There's heterochromatin, which is more electron dense, and then there's euchromatin, which is electron lucent. There is an area that we could consider the nucleolus, and then there's a nuclear matrix. If you were to treat a cell to isolate the nuclei and then treat the nuclei with very harsh chemicals to digest away DNA, RNA, and proteins, there would still be some type of proteinaceous structure left. That would represent the nuclear matrix. Now, as we said, the nuclear envelope consists of two lipid bilayers, so a double membrane. The nuclear envelope has pores that perforate the nuclear envelope at various places. These pores have a very regular architecture, which we're not going to go over here. You may have seen that earlier in your training. But the significance of the pores is they allow small molecules to pass freely in both directions. So molecules less than about 40,000 Daltons can freely pass through the nuclear pores. Larger molecules require specialized localization signals, specialized nuclear localization signals. And the nuclear localization signals in those molecules will interact with some of the structural components in the nuclear pores so that larger molecules can pass. What's shown here is just the nuclear lamina. We're not going to talk about the nuclear lamina now, but the nuclear lamina is almost like a meshwork of protein materials on the inner face of the nuclear envelope, and it gives the nuclear envelope some structure. The nuclear lamina probably also serves as attachment sites for chromatin. That's what's kind of diagrammed here as these chromatin that would be attaching to the nuclear envelope. We'll talk about the nuclear lamina a little bit later in the course when we talk about the cell cycle, because the nuclear lamina changes fairly dramatically during the cell division cycle. You, I think, appreciate that prokaryotic cells don't have nuclei, and you can think of prokaryotic cells as an individual organism, and this cartoon just shows simple prokaryotic organisms, from the simplest, which are mycoplasms, to bacteria, which are a little more complex, to the most complex prokaryotic organisms, the blue-green algae or cyanobacteria. In medical school, we don't talk about cyanobacteria, but you're clearly going to talk about bacteria and mycoplasms, as these organisms play important roles in causing a lot of human diseases. As you know, prokaryotic organisms, very simple. DNA is not bounded by any nuclear membrane. There are ribosomes in these simple mycoplasms. Bacteria are a little more complex, but they still don't have the DNA encased or enclosed in a nuclear envelope. But you may see infoldings in various places of the plasma membrane. These infoldings are called mesosomes, and those function to increase membrane surface area, allowing to have loci on membranes for different types of biochemical activities. The blue-green algae have more inclusions, most notably thylakoid membranes, which are primitive chloroplasts. As you know, prokaryotes divide by binary fission, and eukaryotic cells divide by mitosis or meiosis if one is talking about the production of gametes. Every cell, whether it's a prokaryotic cell or a eukaryotic cell, virtually every cell undergoes glycolysis. And you should recall from biochemistry, glycolysis occurs anaerobically. It's one of the most conserved of all biochemical processes. It probably appeared something like 2.5 to 3 billion years ago. So it's one of the most ancient of all biochemical processes. And in the cartoon, you can see that glycolysis requires a little bit of ATP, but some ATP and NADH are generated from glycolysis. And I'll leave it to you to recall why ATP and NADH are important in the economy of cells. You do know that complete oxidation occurs in the presence of oxygen, and that leads to a very much more level of production of ATP and NADH. So on the cartoon up here would be showing glycolysis with just a little bit of ATP produced, and then complete oxidation in the presence of oxygen, actually termed oxidative phosphorylation, lots more ATP produced. Remember from biochemistry that the end point of glycolysis is pyruvate. Pyruvate going to acetyl-CoA enters the citric acid cycle. Now the entire processes shown on this slide are respiration, and respiration occurs in mitochondria, and the byproducts of respiration, as you remember from biochemistry, are carbon dioxide and water. Mitochondria are a double membrane-bound organelle. Because they have two membranes, mitochondria have multiple organelle compartments. So one could describe the outer mitochondrial membrane, the inner mitochondrial membrane, which folds into cristae. One can describe the space between the inner and the outer mitochondrial membranes, shown here on the cartoon as the white space, and of course the mitochondrial matrix. You should recall from general biology that mitochondria of course contain DNA, but they replicate independently of the cell cycle, so they show non-Mendelian or cytoplasmic or maternal inheritance. This is just a high-power view of a mitochondria, showing very nicely the outer mitochondrial membrane, the inner membrane folding into cristae. You can see where my pointer is, the space between the inner and outer membranes. Here again you can see it over here, the space between the inner and outer mitochondrial membranes. And of course the mitochondrial matrix. The electron-dense deposits are likely calcium deposits, and on this electron micrograph you can also see profiles of rough endoplasmic reticulum showing ribosomes bound to the rough endoplasmic reticulum. This cartoon shows the outer mitochondrial membrane, the inner mitochondrial membrane, the mitochondrial matrix, diagrammatically showing the citric acid cycle, the space between the inner and outer membrane, and then some of the electron transport enzymes that you would have reviewed in biochemistry that are embedded in the inner mitochondrial membrane. You should remember the outer mitochondrial membrane is fairly permeable to most molecules, so it generally allows free passage of material into or across the outer mitochondrial membrane. So one can say that the cytosol and the space between the inner and outer membrane are very similar. But on the other hand, the inner membrane of mitochondria is highly impermeable. Now it does contain, as I suggested, electron transport enzymes, and this sets up a gradient of hydrogen ions across the inner membrane, which provides the energy for oxidative phosphorylation. This process, as you recall from biochemistry, is chemiosmosis. You should remember that hydrogen ions can cross the membrane at the point of these electron transport proteins, and you remember the ATP synthetase embedded into the inner mitochondrial membrane with portions of it pointing to the matrix, and this is where ATP is generated. In addition to the production of ATP, mitochondria are involved in lipid metabolism, and something we don't talk about here, and you probably didn't talk about too much in biochemistry, but mitochondria do play a role in apoptosis, or cell death, as mitochondria contain some of the enzymes that are involved in apoptosis. This is a high-power view of the distal tubule in the kidney. I show this not to expect you to be able to identify this is the distal tubule in the kidney, but I show it because I want to show you mitochondria that are interdigitated within foldings in the plasma membrane of these cells. And this is a very characteristic appearance of cells that are involved in active transport. Wherever you have active transport, you're going to tend to have a lot of mitochondria associated with membranes. You'll discuss this in physiology. We'll talk about this as we go through and talk about some of the different organs later in our course. Here's another interesting view of mitochondria, showing mitochondria with tubular cristae. These mitochondria happen to come from Leydig cells in the testis, and an interesting feature, cells that are involved in the production of steroid hormones show mitochondria with tubular cristae, and obviously the testis produces testosterone. So this is a very characteristic feature of steroid-producing cells, mitochondria with tubular cristae. So steroid-secreting cells will have, in addition to mitochondria with tubular cristae, a very extensive, smooth endoplasmic reticulum. This is because a lot of the enzymes that are involved in lipid biosynthesis are located in the smooth ER, and we'll talk about this a little bit later in the course as well. Now, as we said, mitochondria show maternal inheritance or non-Mendelian inheritance, and I'm not going to spend a lot of time on this here, but you know that if mitochondria develop mutations because of just the fact that mitochondria replicate independently of the cell cycle, over time, just by stochastic events, you can build up cells that may have greater and greater numbers of mutant mitochondria. And if you have cells that have mutant mitochondria, you could have those mitochondria be manifest with a diseased phenotype. And indeed, there are mitochondrial mutations that affect almost every system in the body. We're not going to go over this here, but as you go through your clinical training, you'll come across examples of mitochondrial defects in, for example, the visual system and hearing, and the GI system. As I said, in almost every organ system, there can be mitochondrial mutations that can have significant clinical implications. Compartments of an animal cell, all the compartments tend to be membrane bound, so the cartoon just showing the membrane bound compartments like the nucleus, the ER, the Golgi, mitochondria, maybe lysosomes, peroxisomes, other inclusions, so all the membrane bound organelles. On this side we just show the cytosol just shaded in blue. So the cytoplasm we would consider is the cytosol plus all of the organelles, but not including the nucleus, whereas the cytosol includes everything in the cytoplasm, but not the membrane-bound organelles. Not a big distinction, but if you see those terms, I just want you to understand what they mean. Here is just a cartoon that shows the endoplasmic reticulum, and a little bit higher power micrograph showing rough endoplasmic reticulum, making the point that the rough ER is in continuity with the outer membrane of the nuclear envelope. You should recall the function of the rough ER and the smooth ER. We'll talk about this later in the course, but in a nutshell, rough ER functions for the synthesis of proteins that get exported out of the cell or that become embedded in membrane-bound organelles or in the membranes of membrane-bound organelles. The smooth ER functions in lipid biosynthesis. It functions in detoxification of a lot of foreign substances. And we'll talk a little bit about this later. The Golgi, let me go back for a second. The ER, whether you talk about the rough ER or the smooth ER, is a single membrane-bound compartment. One can contrast that with the Golgi apparatus, which is a series of stacked compartments. So each individual Golgi stack is essentially a separate compartment, and that's shown nicely in the high power electron micrograph. We'll talk about the Golgi in fair detail a little bit later in the course. We're going to talk about the Golgi and the ER essentially together later in the course. We'll talk about an ER Golgi intermediate compartment, and we'll talk about a related compartment, the endosome compartment. In a nutshell right now, we can think of the Golgi as routing or directing traffic of material through the cell in a biosynthetic or an exocytic pathway, whereas the endosome compartment does just the reverse. It directs traffic in an endocytic pathway as material is brought into the cell. And as I said, we'll talk about this later in the course. You are aware that cells have a fairly well-developed cytoskeleton. The cytoskeleton consists of arrays of different protein filaments that give the cell its shape, provide the basis for cells to move. In eukaryotic cells, there are three major types of cytoskeletal filaments: microtubules, which are the largest of the cytoskeletal elements. There are microfilaments, which we know of as actin filaments, and there are intermediate filaments like myosin, and of course there are other intermediate filaments. We will talk about the cytoskeleton in much greater detail later in the course. Now, of course, I don't have to tell you much now about the role of the cytoskeleton in cell division, but clearly the cytoskeleton, in addition to giving the cell its shape and providing an ability for the cell to move, plays an important role in cell division. Microtubules make up the spindle apparatus, which moves chromosomes during mitosis or meiosis. There's actin that plays a role in cell division in the cortical cytoplasm, and then actin and myosin form the contractile ring as animal cells divide. Here, finally, just a comparison of prokaryotic and eukaryotic cells to put some things in perspective for you. So prokaryotic cells, things like bacteria or mycoplasms; eukaryotic cells, think about human cells, but obviously protozoa, multicellular plant cells, or multicellular animal cells. Cell size, obviously eukaryotic cells are generally larger than prokaryotic cells. The genome size is usually larger in eukaryotic cells as compared to prokaryotic cells, but be careful here. Genome size doesn't necessarily relate to complexity of an organism. For example, Drosophila has about 4 picograms of DNA. Humans have about 3.5 picograms of DNA. These are both clearly more complex organisms than an E. coli, which has 0.001 picograms of DNA, but we like to think that humans with 3.5 picograms of DNA are probably more complex than some plants, which can have up to 100 picograms of DNA. So we're not going to talk about why some eukaryotic cells have a lot of DNA, but just understand that generally eukaryotic cells, well, in fact, eukaryotic cells do have a larger genome size than prokaryotic cells. As you are aware, eukaryotic DNA complexes with lots of different proteins, notably histone proteins and non-histone proteins, and arrays itself into multiple linkage groups called chromosomes. I think you discussed that a little bit last semester in biochemistry. Prokaryotic cells, there's a single chromosome, so a single genetic linkage group. The bacterial chromosome is got a much different organization with relation to proteins complexing with DNA. For example, there are no histones associated with bacterial chromosomes. Eukaryotic cells have nucleoli which contain the ribosomal DNA. Prokaryotic cells do not have nucleoli. They obviously do have ribosomal DNA sequences. Prokaryotic cells, as we said, divide by binary fission. Eukaryotic cells dividing by mitosis, and meiosis, the special form of division necessary for gamete production. Something you should recall from biochemistry: eukaryotic cells have larger and more complex ribosomes than prokaryotic cells. So, for example, the eukaryotic ribosomal subunits are 60S for the large ribosomal subunit and 40S for the small ribosomal subunit, as compared to 50S and 30S for the large and small ribosomal subunits, respectively, in prokaryotic cells. And you may recall that S is a measure of the sedimentation coefficient. Prokaryotic cells do not contain any membrane-bound organelles. Clearly, eukaryotic cells have lots of different membrane-bound organelles. In eukaryotic cells, respiration and oxidative phosphorylation occur in mitochondria, in the mitochondrial matrix and across the inner mitochondrial membrane. In prokaryotic cells, this occurs in the cytoplasm and on proteins embedded on the inner face of the plasma membrane. We don't talk about photosynthesis in medical school. Photosynthesis in plant cells occurs in chloroplasts, which are very complex organelles, by the way. They're actually a triple membrane-bound system. Photosynthesis in primitive prokaryotes in the cytosol and on the interface of the plasma membrane. Bacterial cells, they have a cell wall. You'll talk about this in microbiology, but it's not a cellulose cell wall like plant cells have. Eukaryotic cells undergo the process of exocytosis and endocytosis. We'll talk about this in some detail later in the course. This doesn't really occur to any extent in prokaryotic cells. Very extensive cytoskeleton in eukaryotic cells. Almost no cytoskeleton, or not that significant a cytoskeleton, in prokaryotic cells. Eukaryotic cells have cilia and flagella, which makes up the sperm tail. We'll talk about this in some detail later in the course. Prokaryotic cells don't have cilia. They do have flagellum, and you'll see this in bacteria, but the flagella is not at all like a eukaryotic flagella. There's a particular protein in prokaryotes called flagellum. This is not a microtubule-based structure like in eukaryotic cells. And then finally, protein synthesis can be inhibited by various types of drugs, and this will be foundational in pharmacology. Different types of drugs can affect protein synthesis differently in eukaryotic cells versus prokaryotic cells. So, for example, antibiotics like chloramphenicol do not affect protein synthesis in eukaryotic cells, but they do affect protein synthesis in prokaryotic cells. Likewise, cyclohexamide does not block protein synthesis in prokaryotes, but it does block protein synthesis in eukaryotes. So just a point to keep in mind that different drugs will affect eukaryotes and prokaryotes differently.